Hi everyone, I'm Lyena, a bachelor student in Kyushu University. Today, I'd like to talk about a protein, gluten, and the genetic technology. First, I start with bread. Do you like eating bread? Freshly baked rolls look like fluffy clouds in the blue summer sky. What makes bread so attractive is the protein called gluten found in wheat, rye, and barley. But in patients with a serious autoimmune disease called celiac disease, the gluten damages the small intestine. More patients with gluten hypersensitivity who have milder symptoms are also unable to eat foods containing gluten. Most gluten-free bread uses rice flour or potato starch instead of wheat flour, and has a different taste and texture from wheat bread. But recently, there has been a report of a genetically engineered method of producing wheat that is low in gluten content but still contains other proteins that produce the bread's unique taste and elasticity. Genetically modified crops are the object of intense debate around the world, and some countries such as France and Germany ban their cultivation by law. The biggest concern is inserting DNA from one species to another, says plant bioengineer Francisco Barro, a plant scientist at the Institute for Sustainable Agriculture in Spain. To avoid these gene crossovers, Barro and colleagues used the gene editing technology CRISPR-Cas9 to excise specific genes from the wheat genome. CRISPR technology is a simple yet powerful tool for editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. Its many potential applications include correcting genetic defects, treating and preventing the spread of disease, and improving crops. The Cas9 protein is an enzyme that cuts foreign DNA.
The target was a protein called alpha-gliadin, which is deemed to be the main component of wheat gluten that causes troublesome effect on the immune system. The team designed a molecule that directs the Cas9 enzyme, which acts as a scissors for genome editing, to excise 35 of the 45 genes involved in alpha-gliadin production in wheat. An experiment in which the modified wheat was examined on a petri dish weakened the immune response by 85%, reported in the September 2018 Plant Biotechnology journal. And it's from another thesis. One of the challenges with current gluten-free products is that they have a different flavor and texture. Barro's team has collaborated with a baker in Spain to use their low-gliadin RNAi line of wheat to create bread with flavor and aroma indistinguishable from standard wheat bread. Celiac patients have been able to eat this bread and report on its quality. First, this is a very hot topic, a very nice example of using biotechnology to help people. Everyone knows celiac people, and celiacs know that bread is a very difficult product, that gluten-free bread is not as good as other gluten-free products. I think that for a celiac to enjoy good bread made of wheat with the taste of wheat, the aroma of wheat, it will be something very amazing, and we are getting closer. Second, most papers report the use of gene editing technology being limited to only a few genes. In our work, we report the simultaneous mutation of at least 35 different genes in bread wheat. And this is something really outstanding, he said.
While seeing their references, listen to me please. Finally, this is in my opinion. I study developmental disabilities and fibromyalgia syndrome. And I realize both diseases have a large relationship with gluten, so I hope this technology will improve these in the future.
Thank you.
Quería volver a